Welcome to the Research Highlights video series. These videos are intended to provide a summary of some of the research being conducted by members of the Hopkins Group at the University of Waterloo. This video highlights some of our work on determining the structures and properties of amino acid containing clusters. For a more in-depth description of the structures and properties of clusters which contain phenylalanine derivatives, please read our publication in the journal Physical Chemistry Chemical Physics. Our goal in this study was to determine the structures of gas phase clusters of trimethylamine and 3-fluoromethyl-phenylalanine, and to try to influence cluster fragmentation via vibrational mode selective excitation. Using our basin hopping technique, we mapped the potential energy landscape of the protonated TMA 3-fluoromethylphenylalanine cluster. This mapping identified three cluster binding motifs: a canonical charged solvated motif with protonation on the TMA, a zwitterionic motif with protonation on the TMA, and a TMA bridging motif in which the proton is located in a bridging point between the TMA and the nitrogen and carbonyl oxygen centers of the amino acid. By representing our topological map of the potential energy surface as a disconnectivity graph, we are able to identify isomerization and proton transfer pathways for the cluster. The disconnectivity graph also shows that there are fairly large barriers to conformational change and tautomerization, and we see a deep secondary funnel on the potential energy landscape, which is associated with the TMA bridged binding motif. These calculations suggest that several different structures from all three binding motifs should be kinetically trapped in a gas phase ensemble, so we should observe a rich vibrational spectrum. When we conduct infrared multiple photon dissociation studies of the cluster, we observe very interesting behavior. We see that the cluster dissociates to form either protonated TMA or the protonated amino acid. What's more, the IR spectra observed in these two product channels are different. If we focus on the carbonyl stretching region at 1750 reciprocal centimeters, we see three peaks. Since there is only one carbonyl group in the cluster, the three observed carbonyl bands must correspond to at least three different cluster structures. By comparing experimental results with quantum chemical calculations, we see that canonical charged solvated structures produce protonated TMA when they fragment. The zwitterionic and TMA bridged species dissociate to produce the protonated amino acid. Interestingly, for excitations below 1300 reciprocal centimeters, all three binding motifs access both product channels. Taken as a whole, this shows vibrational mode selective behavior. We can dial up a particular product distribution by exciting specific vibrational motions. At lower wave numbers, all three binding motifs exhibit similar spectra. If we excite the canonical structures to access the protonated TMA products, but undergo proton transfer to yield a zwitterionic structure en route, the zwitterionic species can continue absorbing IR radiation via its near-resonant vibrational transition such that the protonated amino acid is generated. If, on the other hand, we excite the canonical structures via the carbonyl stretch at 1740 reciprocal centimeters, and tautomerization occurs to form a zwitterionic species, there is no near-resonant zwitterion transition to continue absorbing infrared radiation. This photo-induced transparency effectively removes the protonated amino acid product channel. Our research team is currently exploring the use of IR mode selective behavior to control chemistry in other chemical systems. For more information about the Hopkins Group members and the research that we conduct, please visit the Research Group website, which is hosted by the University of Waterloo Department of Chemistry. Thank you for your time and your interest in our work. See you next time.